Basics of Medical Biochemistry. First edition, 2023. Chapter 17. Chemistry of Nucleic Acids, DNA Replication, DNA Damage and Repair. Part 1 of 3. By Professor Dr. Muhammad Jawad. MBBS, Tao, M. Philosophy, Ph.D., Dip M.E. Head of the Biochemistry Department-Fazaia Ruth Pfau Medical College, Air University-PAF Base Faisal-Karachi-Pakistan. Ex-Professor of Biochemistry, Qassim University Kingdom of Saudi Arabia. Ex-Professor, Head of the Biochemistry Department and Vice Principal. Liaquat College of Medicine and Dentistry and Fatima Jinnah Dental College, Karachi, Pakistan. Chapter 17. Chemistry of Nucleic Acids, DNA Replication, DNA Damage and Repair. 17.1 Introduction. Nucleic acids are non-protein nitrogenous polynucleotide biopolymers found in all living cells, even in bacteria and viruses. They are regarded as essential substances of the genes and act as the main information-carrying molecules of the cell. They direct the process of protein synthesis and hence determine the inherited characteristics of every living organism. The two main classes of nucleic acids are deoxyribonucleic acid, DNA, and ribonucleic acid, RNA. Nucleic acids were first demonstrated in the nuclei of pus cells in 1868 and sperm heads in 1872 by Swedish physician Friedrich Miescher, and at that time they were named nuclein. Basic concepts. A DNA molecule is composed of two polynucleotide strands, double strands, that spiral around to form a double helix, while an RNA molecule is a single polynucleotide chain, single strand. DNA is present in the nuclei as parts of the chromosomal structure and mitochondria of all cells. RNA is a single strand of a polynucleotide, exists in many forms and is found throughout the cell. DNA can store, express and transmit genetic information. It can also undergo mutation, which is essential for the evolution of new varieties and species. Eukaryotic DNA occurs in combination with histone, a type of nucleoprotein. Under the electron microscope, chromatin shows dense spherical particles composed of DNA wound around a histone molecule called nucleosome. Prokaryotic DNA exists as a part of nuclear material being scattered in the cytoplasm to form a structure called nucleoid. RNA is mainly concerned with the synthesis of proteins. RNA also acts as an enzyme and may represent the genome instead of DNA as in RNA viruses. The study of nucleic acids is playing a significant role in cancer research. Inborn errors of purine and pyrimidine metabolism result in various diseases like gout and orotic aciduria respectively. Genetic engineering or recombinant DNA technology is considered a breakthrough in the treatment of genetic diseases. By this technique, a large variety of useful substances like hormones, example insulin, antigens, antibodies, and many other therapeutic agents have been produced. Artificial cloning is now possible through recombinant DNA technology. Genetic testing is a type of medical test that identifies changes in chromosomes, genes, or proteins. The results of a genetic test can confirm or rule out a suspected genetic condition or help determine a person's chance of developing or passing on a genetic disorder. It provides the foundation of gene therapy. Genetic counseling is a process to evaluate and understand a family's risk of an inherited medical condition. DNA fingerprinting or genetic fingerprinting, also known as DNA profiling, is a molecular genetic method that enables the identification of individuals using hair, blood, semen, or other biological samples, based on unique patterns, polymorphisms, in their DNA. It is also used to establish paternity. The Human Genome Project was accomplished in 2003. It refers to an international effort of 13 years to discover all the estimated 99.99%, 30,000 to 35,000 human genes located on 23 pairs of chromosomes in humans with 99% accuracy. This project also determined the complete sequence of 3 billion DNA subunits. A genome library refers to the record of a copy of genetic information of species in a preferred environment. 17.2 Structural organization of nucleic acids. Nucleic acids are made up of a large number of nucleotides. Each nucleotide is composed of a nitrogenous base purine or pyrimidine. 2-pentose sugar is deoxyribose in DNA and ribose in RNA. It is attached by its C1 to N1 of pyrimidine or N9 of a purine by a glycosidic bond. 3-phosphate group act as the backbone by attaching nucleotides to one another in the polynucleotide chain of nucleic acid. Nitrogenous bases, these are nitrogen-containing heterocyclic, aromatic compounds. They include purine and pyrimidine derivatives. Purines. A purine is composed of a 6 and a 5-member nitrogen-containing ring, fused together. It is formed by the union of the pyrimidine ring with the imidazole ring. Important types of purines include adenine, A, i.e. 6-aminopurine, and guanine, G, i.e. 2-amino-6-oxypurine, both of which are found in DNA and RNA. Other less common purine derivatives include hypoxanthine, 6-oxypurine, and xanthine, 2,6-deoxypurine. Pyrimidines, these include cytosine, C, thymine, T, and uracil, U. Cytosine is found both in DNA and RNA, whereas thymine is found only in DNA and uracil is found only in RNA. Figure 17.1 Purine, pyrimidine, and their subtypes. There are some minor or unusual nitrogenous bases also found occasionally in nucleic acids such as methylpurine, methylpyrimidine, pseudoridine, etc. 2-pentose sugars, D-ribose and D2-deoxyribose, are found in RNA and DNA respectively. Figure 17.2 Pentose sugars found in nucleic acids, ribose, left, and deoxyribose, right. 3-phosphate group, it is attached to C5 of the sugar below and C3 of the sugar above in the polynucleotide chain of nucleic acid. 17.2.1 Nucleosides. Pentose sugar plus nitrogenous base. A nucleoside is formed by the combination of a purine or pyrimidine nitrogenous base with pentose sugar, ribose or deoxyribose. There is a glycosidic linkage between C1 of pentose sugar and N1 of pyrimidine or N9 of purine. The nucleoside of RNA is called a ribonucleoside, while the nucleoside of DNA is known as a deoxyribonucleoside. The pentose sugar present in them is ribose and deoxyribose respectively. Purine nucleosides end in sine as adenosine and guanosine. Pyrimidine nucleosides end in dine as thymidine, cytidine, and uridine. The syn and anticonformers of adenosine differ in orientation about the N-glycosidic bond. Figure 17.3 Types of nucleosides. 17.2.2 Nucleotides. Pentose sugar plus nitrogenous base plus phosphate or nucleoside plus phosphate. Nucleotides are phosphoric acid esters of nucleosides. There are two types of nucleotides, i.e., ribonucleotide and deoxyribonucleotide, found in RNA and DNA respectively. Ribonucleotides are composed of phosphorylated ribose with any purine, adenine or guanine, and cytosine or uracil variety of pyrimidine. Deoxyribonucleotides are composed of phosphorylated deoxyribose with any purine, adenine or guanine, and cytosine or thymine variety of pyrimidine. It is to be noted that uracil is only found in RNA, whereas thymine is only found in DNA. Figure 17.4 Types of nucleotides. In the case of ribose, there are three possible positions, C2, C3, and C5, while in deoxyribose there are only two possible positions, C3 and C5, to form ester linkage with phosphoric acid. To differentiate the atoms of the sugar from the nitrogenous base, the position number of the sugar is followed by a prime. As 5O of the sugar is most commonly esterified with phosphoric acid, that's why 5 is written without its prime. For example, adenosine 5-monophosphate is usually written as 5-monophosphate. Table 17.1 Nitrogenous bases and their respective nucleosides and nucleotides. Nitrogenous base. Ribonucleoside. Ribonucleotide. Deoxyribonucleoside. Deoxy. Ribonucleotide. Adenine, A. Purine. Adenosine. Adenosine monophosphate, AMP, or adenylic acid or adenylate. Deoxyadenosine. Deoxyadenosine monophosphate, DAMP, or deoxyadenilic acid or deoxyadenilate. Guanine, G. Purine. Guanosine. Guanosine monophosphate, GMP, or guanilic acid or guanylate. Deoxyguanosine. Deoxyguanosine monophosphate, DGMP, or deoxyguanilic acid or deoxyguanylate. Cytosine, C. Pyrimidine. Cytidine. Cytidine monophosphate, CMP, or cytidilic acid or cytidylate. Deoxycytidine. Deoxycytidine monophosphate, DCMP, or deoxycytidilic acid or deoxycytidylate. Thymine, T. Pyrimidine. Not found in RNA. Not found in RNA. Deoxythymidine or thymidine, without deoxy. Deoxythymidine monophosphate, DTMP or only TMP, or thymidilic acid or thymidylate. Uracil, U. Pyrimidine. Uridine. Uridine monophosphate, or uridilic acid / or uridylate. Not found in DNA. Not found in DNA. Thymine occurs only in DNA and uracil occurs only in RNA, but its deoxy derivative, i.e., deoxyuridine monophosphate, DUMP, has an important role in the biosynthesis of thymine. Some of the synthetic purine and pyrimidine analogues act as immunosuppressors to control immune response after organ transplant, as well as therapeutic agents for the treatment of cancer and AIDS. 17.2.3 Biologically important nucleotides. Following are some other biologically important nucleotides. 1. ADP, ADP, and AMP. A mononucleotide having single phosphate can be further phosphorylated to D and triphosphate. For example, successive phosphorylation of AMP brings about the formation of ADP and ADP. The main role of ADP is to provide energy. It is regarded as the energy currency of the biological system. It can store and transport chemical energy within the cells. A large number of life processes need ADP, such as the synthesis of biomolecules, muscle contraction, membrane transport, transmission of nerve impulses, motility of spermatozoa, etc. Figure 17.5 Structures of AMP, ADP, and ADP. 2-cyclic adenosine monophosphate, CAMP, or 3,5-cyclic adenosine monophosphate. It acts as a second messenger and is used for intracellular signal transduction in many biological processes. For example, it is involved in the activation of inactive glycogen phosphorylase B to its active form glycogen phosphorylase A by adrenaline. It is a nucleotide synthesized within the cell from adenosine triphosphate, ADP, by the enzyme adenyl cyclase. It is inactivated enzymatically to 5-AMP by hydroxylation with a group of enzymes called phosphodiesterases. Figure 17.6 Structure of cyclic AMP, CAMP. 3-active sulfate, adenosine 3-phosphate 5-phosphosulfate. It is derived from ADP on reaction with H2SO4 in the presence of magnesium ions. It is required for the addition of sulfate groups in many compounds. 4-active methionine. It is S-adenosylmethionine, SAM or SAME, derived from ADP and methionine. It has a role in the transfer of one carbon moiety, example methyl, CH3, group in many reactions. It has been used in alternative medicine as a likely effective aid in reducing the symptoms of depression and osteoarthritis. 5-GTP and GDP, these are guanosine-derived nucleotides. GTP is involved in energy transfer within the cell. It is required for many biological processes like oxidation of succinyl-CoA in the citric acid cycle, protein synthesis, purine synthesis, formation of CAMP, rhodopsin cycle in the eye, gluconeogenesis, etc. GTP and GDP are also involved in the activity of membrane receptors belonging to the G family. A molecule of GTP is generated in the citric acid cycle, where it is readily converted to ADP by the enzyme nucleoside diphosphate kinase. 6-cyclic guanosine monophosphate, CGMP. It is a cyclic nucleotide derived from guanosine triphosphate, GTP. It acts as an important intracellular second messenger that mediates multiple tissue and cellular responses. Its most likely mechanism of action is the activation of intracellular protein kinases in response to the binding of membrane-impermeable peptide hormones, example atrial natriuretic peptide, to the external cell surface. 7-cytidine triphosphate, CTP. It is a pyrimidine ribonucleoside 5-triphosphate. It acts as a substrate in the synthesis of RNA. It takes part in the synthesis of lecithin by forming CDP-choline. CMP-sialic acid is found in salivary glands and is involved in the synthesis of salivary mucin. It facilitates and improves the identification of the infarct core and has the potential to identify tissue at risk. 8-uridine diphosphate glucose, UDP glucose. It is involved in glycosyltransferase reactions in metabolism, particularly in glycogen synthesis. It can be metabolized into UDP galactose and UDP glucuronic acid. 9-uridine diphosphate galactose, UDP galactose. It takes part in the conversion of galactose to glucose. 10-uridine diphosphate glucuronic acid, UDP glucuronic acid. It is utilized in the conjugation and detoxification of bilirubin, benzoic acid, steroids, menthol, etc. It is also used in the synthesis of hyaluronic acid, heparin and other mucopolysaccharides. 17.3 Structure of DNA.
In the molecule of nucleic acid, nucleotides are joined at the C5 and C3 of each sugar backbone by a phosphodiester bond. The sugar-phosphate linkages form the backbone of the polymer to which the variable bases stick. The nucleotide polymer has a free phosphate group attached to the 5 position of sugar and a free 3-hydroxyl group. This is called the polarity of the DNA molecule. The base sequence of the polynucleotide is always read and write from the 5 to 3 direction, example 5-GCAT3. Figure 17.7 Formation of the polynucleotide to represent the primary structure of nucleic acid. DNA is a double-stranded helix composed of 2 polynucleotide chains as proposed by Watson and Crick, 1953. The nitrogenous bases found in DNA are adenine, A, guanine, G, thymine, T, and cytosine, C. Their order is important in determining the genetic information of the molecule. Sugar is deoxyribose with a phosphate group. 4 deoxyribonucleotides found in DNA are DAMP, DGMP, DCMP, and DTMP or simply TMP.
Figure 17.8 Complementary base pairs in DNA.
DNA is present in all cells and virtually restricted to the nucleus. The amount of DNA in somatic cells, body cells, of any given species is constant, like the number of chromosomes. The DNA content of gametes, sex cells, is half that of somatic cells. The human diploid cell contains 3.2 times 109 base pairs in DNA. The length of fully stretched DNA found in the chromosome of a single diploid human cell is 2 meters and but the total length of the whole DNA of all 46 chromosomes is just 200 nanometers. The DNA helix is 2 nanometers thick. It means that DNA occurs in a highly condensed form. The total amount of DNA estimated in a diploid human cell is 17 picograms.
17.3.1 Primary structure of DNA. It describes the linear sequence of deoxyribonucleotide units. 17.3.2 Secondary structure of DNA. The secondary level of the structure of DNA explains its double-stranded helix formation. In the helical structure of DNA, the purine and pyrimidine bases are found on the inside while sugars and phosphates are present on the outer side. The 2 strands of DNA run in opposite directions, anti-parallel, and coiled around each other in a double helix. These are connected by hydrogen bonds, individually weak but collectively strong. Adenine on one strand binds to thymine, A equals T, of the opposite strand by 2 hydrogen bonds. Guanine on one strand binds to cytosine, GC, of the opposite strand by 3 hydrogen bonds, which is called base pairing and base complementarity. Thus, the sister strands are complementary but not identical. The higher number of hydrogen bonds in GC pairing confers sequence-specific annealing properties. Erwin Chargaff, 1950–51, studied DNAs from various sources and analyzed the distribution of purines and pyrimidines in them. He proposed Chargaff's law and concluded that the distribution of nitrogenous bases varied among species. But the total purines, A and G, and the total pyrimidines, T and C, are always equal. Moreover, %A equals %T, and %G equals %C. Mammalian DNA has 60% at pairs and 40% GC pairs. In the B form, most common type, of DNA molecule, the helix makes a turn at every 3.6 nanometers, and the distance between two neighboring base pairs is 0.36 nanometers and each base is rotated at 36 degrees in relation to the previous base. Hence, there are about 10.5 base pairs per turn. The intertwined strands make two grooves of different widths, referred to as the major groove and the minor groove, which may facilitate binding with specific proteins. One strand in DNA is a coding strand and the other is the template. The template strand of DNA is copied during RNA synthesis. It is sometimes referred to as the non-coding strand. The coding strand has a sequence of bases similar to the sequence of the RNA transcript, but having uracil in place of thymine. Figure 17.9 Secondary structure of DNA. DNA also exists in some other forms such as A, Z and H forms. A form of DNA is produced by the crystallization of B form after its dehydration. It is a right-handed helix, having 11 base pairs per turn and its thickness is 2.6 nanometers. The Z form of DNA is slenderer, elongated and left-handed. It has 12 base pairs per turn. Its major groove is less prominent while the minor groove is constricted and deep. Its involvement has been noted in gene expression and recombination. The H form of DNA is found in polypurine or polypyrimidine tracks in the form of a triplex. In some situations, DNA can also exist as tetraplex, having 4 strands. It is due to a non-Watson-Crick pairing called Hoogsteen pairing after the name of the scientist who discovered it. Another form is satellite DNA as it is obtained as sediments separated from DNA upon ultracentrifugation. It represents simple sequence repeats, SSRs, that constitute 30% of the total human DNA. It is functionally inactive. Figure 17.10 Structure of A, B and Z DNA. Table 17.2 Characteristics of A, B and Z DNA. Feature: A DNA, B DNA, Z DNA. Helix sense: right-handed, right-handed, left-handed. Base pairs per turn: 11, 10.5, 12. Rotation per base pair: +3 degree, +3 degree, 30 degree. Base pair tilt: 20 degree, -6 degree, 7 degree. Axial rise: 0.26 nm, 0.34 nm, 0.45 nm. Diameter of helix: 2.3 nm, 2.0 nm, 1.8 nm. Glycosidic bond conformation: anti-for pyrimidine, syn for purine. Major groove: present, absent or convex shaped. Minor groove: present, deep cleft-like. Source: Encyclopedia of Life Sciences, 2002. Macmillan Publishers Ltd., Nature Publishing Group /www.els.net. 17.3.3 Tertiary structure of DNA. At the tertiary level of structure, double helix wrapped around histone to form nucleosome, 10 nanometers. The double-stranded DNA wraps around a basic protein, histone octamer core to form the nucleosome. Hist1's octamer is H2A, H2B, H3, H4, 2. Because of their positive charge, they form ionic bonds with negatively charged DNA. Hist1s are rich in lysine and arginine residues. Nucleosomes are linked by a DNA linker wrapped around H1. The function of nucleosomes is to condense and stabilize DNA. Nucleosomes appeared as beads and strings in a long series. The association of histone octamer with DNA prevents its hydrolysis by an enzyme nuclease. During the replication of DNA, parental hist-1s remain associated with one of the parental DNA strands. In this way, parental hist-1s are conserved.
Figure 17.11 Tertiary structure of DNA.
At further higher order of structural organization, nucleosomes are supercoiled to form the chromatin filaments, 30 nanometers. The chromatin filaments are supercoiled to form the chromatids and then the chromosomes.
Figure 17.12 Structural organization of DNA up to chromosome. 17.3.4 Mitochondrial DNA. It is a small circular molecule that carries genetic coding for the synthesis of mitochondrial proteins and also for the synthesis of rRNA found in mitochondria. It makes up approximately 1% of total cellular DNA. At the time of fertilization, the mitochondria of the sperm are excluded. As a result, mitochondrial DNA is believed to be inherited from the mother only. Mitochondrial DNA is more delicate and susceptible to mutation than nuclear DNA. This results in a variety of neurological disorders, myopathies and even diabetes mellitus. It is to be noted that human mitochondrial DNA read UGA as a codon for tryptophan rather than as a stop signal. Furthermore, AGA and AG are read as stop signals rather than as codons for arginine. Similarly, AUA is read as a codon for methionine instead of isoleucine. The genetic codes of mitochondria differ from that of the rest of the cell because mitochondrial DNA encodes a distinct set of tRNAs. 17.3.5 Nucleoid and plasmid. The chromosome of prokaryotic organisms exists as a single, double-stranded, supercoiled circular structure. It is associated with hist-1s like protein and RNA in a condensed form known as the nucleoid. Most species of bacteria also contain small, circular, extrachromosomal DNA called a plasmid. They carry genetic information and these are used as a vector in recombinant DNA technology. 17.4 Properties of DNA. 17.4.1 Denaturation of DNA. It is the separation or unwinding of two strands of DNA. It usually occurs at high temperatures and extremes of pH, above 10 and below 4. The denaturation may also be carried out by the effect of strong alkali, or by some physical factors. The temperature at which DNA is half denatured is called the melting temperature, Tm. Tm is greatly influenced by the base composition of DNA. The Tm is higher for the DNA which is rich in GC pairs than for the DNA which has more at pairs. It is because of the presence of 3 hydrogen bonds between G and C while there are only 2 hydrogen bonds between A and T. The Tm of mammalian DNA is 87 degrees C, and it has about 40% GC pairs. As a result of denaturation, the molecular mass becomes half, and viscosity and optical activity are decreased. The absorbance of denatured DNA at 260 nanometers is increased. This is called the hyperchromic effect. Certain chemical agents like urea and formamide facilitate denaturation by disrupting hydrogen bonds among base pairs.
17.4.2 Renaturation, annealing, of DNA. The denatured DNA can be renatured after the removal of the harmful effects. The melted DNA which is denatured by heat is renatured by careful slow cooling. 17.4.3 Genes, introns, and exons, segments of DNA specifying the synthesis of a functional protein or RNA product are called genes. About 30% of DNA is composed of genes but only 1.5% is involved in coding while the remaining 28.5% act as non-coding. The coding part of the gene is known as an exon whereas the non-coding region is termed an intron. Both introns and exons can undergo transcription but mRNA formed by introns is completely removed by a set of splicing enzymes. Pseudogenes have sequences that look like genes but are never expressed. 17.4.4 Transposons. These are repeating sequences of several types that can move from one point to another in the genome. Due to this shifting property, they are also known as transposable elements. They constitute 45% of DNA. DNA transposons can be used to introduce a piece of foreign DNA into a genome. They act as useful tools to analyze the regulatory genome, study embryonic development, identify genes and pathways implicated in the pathogenesis of diseases, and even contribute to gene therapy. The following 4 types of transposons have been identified. I long interspersed elements, lines 21%. 2 short interspersed elements, sines 13%. 3 retrovirus-like elements, 8%. 4 remnants of transposons, 3%. 17.4.5 miscellaneous sequences, they represent 25% of the total genome. They include large segmental duplications, SDS, 5% that appear more than once in a different location, and simple sequence repeats, SSRs, 3%. The remaining 17% is comprised of complex and not identified sequences as remnants of transposons. Table 17.3 Distribution of different regions on DNA. Genes 30%. Transposons 45%. Miscellaneous 25%. Exons 28.5%. Introns 1.5%. Long interspersed elements, lines, 21%. Short interspersed elements, sines, 13%. Retrovirus-like elements 8%. Remnants of transposons 3%. Large segmental duplications, SDS, 5%. Simple sequence repeats, SSRs, 3%. Remnants of transposons 17%. 17.4.6 Hybridization. Hybridization reactions can occur between any single-stranded nucleic acid chains. It occurs when a mixture of DNA and RNA is heated up to Tm to form single strands, and then cooled RNA can hybridize, form a double helix, with DNA that has a complementary nucleotide sequence. In molecular genetics, by the technique of hybridization, single strands of DNA from 2 different species are allowed to join together to form hybrid double helices. 17.4.7 Digestion and ligation. DNA molecules can be digested into smaller fragments by some enzymes such as endonucleases. These enzymes can cut the strand of DNA at specific sites. In reverse, a DNA molecule can be reconnected again by other enzymes called as ligases in a process called ligation. 17.5 Junk DNA. The term junk DNA refers to the non-coding regions of DNA. Non-coding regions of DNA have no apparent function. Codes found in DNA are used to create proteins in the cell. However, the amount of DNA contained inside each cell is vast, and not all of the genetic sequences present within a DNA molecule code for a protein. Exons are the regions of DNA that contain the code for producing the polypeptide molecules that make up protein. Each exon codes for a specific portion of the complete protein. In humans and some other species, the exons are separated by long regions of junk DNA. It is suggested that over 98% of the human genome does not encode protein sequences, including most sequences within introns and most intergenic DNA, while 20% of a typical prokaryote genome is non-coding. Much of the junk DNA consists of multiple repeats of lengths of DNA that appear to serve no useful function other than that they provide indicators of human phylogeny. It has recently been suggested that there may be a relationship between the introns and proteins, and that the introns influence the expression of an exon. Junk DNA is even more conserved than protein-coding regions of the DNA in humans and other mammalian species. The extent of conservation indicates that there is some function for junk DNA that remains to be determined. Currently, a large number of investigations are underway to establish some possible role of junk DNA in the field of molecular biology. It means that junk DNA is not junk in the real sense. Exercise questions. For MCQ 5 to 8 see Part 2 and for MCQ 9 to 12 see Part 3 of this chapter. MCQ 1. Which of the following bonds is found between the nitrogen base and pentose sugar in the nucleotide? Ester. Glycosidic. Hydrogen. Ionic. Peptide. MCQ 2. A 21-year-old man is presented with marked sensitivity to sunlight, ultraviolet, with subsequent formation of multiple skin cancers and premature death. Physical examination and investigation diagnosed him with a case of xeroderma pigmentosum. Which of the following DNA repair mechanism is mutated in this patient? Base excision repair, BER. Direct repair, DR. Double strand break, DSB, repair. Mismatch repair, MMR. Nucleotide excision repair, NI. MCQ3. During DNA repair, the created gap is filled by which one of the following enzymes? Endonuclease, hexanuclease, ligase, polymerase, topoisomerase. MCQ4. Which of the following anticodons would be on the tRNA for the 5AGC3 codon of respective messenger RNA? 3 August 5th. 3 UCG5. 5 AGU3. 5 UUC3. 5 August 3rd. End of Part 1 of 3 of Chapter 17. Thank you.